Thank you, Alan, for those very kind words, and I'm very pleased everyone's very happy. So I'm going to try and convince you today that psychosis or schizophrenia has an element of pathology which is associated with disturbed protein regulation, and that it's probably an imbalance of homeostasis. And probably all illnesses are an imbalance of homeostasis, but I want to, especially towards the end of the talk, tell you that I think it's a disturbance of proteostasis or protein regulation. I'm going to continue Carmina's theme about inflammation. I'm going to talk about the innate immune system, including some work I did while I was in San Diego on the effect of methamphetamine on the brain. And as we know, people with methamphetamine have high rates of psychosis. I'm going to also talk about the related Wnt system. Wnt is a critical pathway in the development of the brain, and it also is active in the adult brain, and we know it goes wrong in Alzheimer's disease. I showed it goes wrong in HIV dementia, and there is now increasing evidence that it's abnormal in schizophrenia. And then I'm going to move on to the work we've been doing the last 3 or 4 years on the ubiquitin proteasome system. So I think it's all about proteins, and each cell has to regulate the protein content in it, and the UPS, or ubiquitin proteasome system, is the way in which proteins are regulated and broken down. And then finally, I'll talk about some work we're currently doing on iron, and how that may well be important in psychosis. So homeostasis is that process which is critical to the normal function of cells all the way up to the full organism. And it was first talked about in 1878 as the need to maintain a stable environment so that biological processes could occur despite the environment. And in 1929, Walter Cannon actually termed the term homeostasis, and it's now been expanded to include behavioural responses as well. And, you know, it's about keeping things in acceptable range, whether it's blood pressure, insulin, lipids, etc., and also neurotransmitters. We can think of 3 levels of homeostasis. Systemic homeostasis, that's variables that regulate the whole organism, such as blood glucose, oxygen, pH, etc. And then there's talk about tissue homeostasis, not so well understood, but that's going to include cell number, cell size, architecture, and things like the maintenance of the extracellular matrix, which is very important in the brain. And then we could think about cellular homeostasis. Again, that's the mechanism by which the cell volume and cell number is maintained, as well as the pH, the membrane potential, etc. And, most importantly, the ability to regulate misfolded or damaged proteins. That's critical to the cellular health of all cells. I'll explain about that process later on. While most cells can effectively recognize homeostatic disruption, there are a few cells in the body which are experts at this. And this is tissue-resident macrophages, or in the brain, microglia, and also mast cells and sensory neurons. And microglia monitor the extracellular matrix in order to make sure that everything is fine. But they also respond not only to infections, but hypoxia and metabolic stress. And when they recognize that something wrong is going on, they produce cytokines, as Carmina talked about, in order to alert other microglia and other cells that things are not as they should be. And this tissue-level stress response may have features of inflammation without the features of classic acute inflammation, where you get exudate formation and neutrophil recruitment. So I'm talking more about this para-inflammation, and I think that's probably what is probably coming to be seen as more important in the development of mental illness, as we heard with depression. So are there any changes to neurons in schizophrenia? Well, we know that there's probably a decrease in the density of GABAergic neurons. This was described a couple of decades ago by Francine Bennis. We also know there's a decrease in neuronal size, and it's been demonstrated in several regions of the brain, the hippocampus, in the cortex, in the cingulate, entorhinal, motor prefrontal cortex, and deeper in the brain in the substantia nigra and locus coeruleus. The most robust anatomical findings that occur in the brain in schizophrenia are changes to synapses and dendrites. And all the markers that you can think of show the reduction in dendritic markers and synaptic markers. So synaptophysin, which is a presynaptic protein, is decreased. SNAP25, another synaptic protein, is decreased, as is GAP43. And complexin 2. These are found in excitatory neurons. And MAP2, this is a protein that is the skeleton for all dendrites. That's also been shown to be decreased in neurons in the brain in schizophrenia. We always concentrate on neurons, so neuronal changes are the be-all and end-all of any pathology. Well, it's not just about neurons. Glial cells are also very important in the brain, and they outnumber neurons by 10 to 1, and they can be astrocytes, oligodendrocytes, and microglia. Now, in this here, that blue box shows you the typical appearance of glial cells in the brain. The red box shows you a typical appearance of a pyramidal neuron, and then the two brown photographs here are photographs of microglia. So this is looking for a protein called IBA1, which is only produced in microglia. On the left-hand side, you can see a typical microglia that's resting. It's got lots of processes going out into the tissue, and then in this case, those processes have been retracted, and this is the typical appearance of an activated microglia, where it's assumed that there is some problem going on in the brain, and it's now producing cytokines. These photographs are from a study I did on autism, and this was the typical appearance of microglia in autistic brains. But I'm not going to talk about autism today. We and others have shown that there are specific changes in glia in the brain of schizophrenia, including the anterior cingulate and also in the dorsolateral prefrontal cortex, which is a region of the brain which we know goes wrong in schizophrenia. And that the glial cell changes include microglia, astrocytes, and oligodendrocytes. And what a few studies around have shown an increase in the density of microglia in the prefrontal and auditory associational cortex. Again, carrying on this idea, there is an inflammatory component to a psychiatric disorder such as psychosis. So when you think about cellular homeostasis, what do cells need to do? Well, they need to make their DNA into RNA and eventually into protein. It's a protein that makes the cell work. They also need to be metabolic. They need to produce energy by ATP. And the problem with this is you end up having to have a reaction with iron, which can produce oxidative stress and oxidative radicals. And I'll come back to iron right at the end. They also need to respond to changes in the environment, such as low nutrition environments, also to be able to recognize and combat any infectious pathogen as well. And then I come back to this idea that they need to be able to sense and remove damaged proteins or proteins that have gone to the end of their life. Certain cells, such as neurons, need to be able to transport molecules and proteins down the dendrites to the synapses, and also certain cells need to be able to reproduce. And again, we need to be able to maintain the cellular structure, either by UPS or autophagy, in order to remove damaged proteins. So here is a diagram of the life of a protein. So DNA is made into RNA, which then results in protein synthesis in the ribosomes. You get this unfolded protein, and that protein has to be folded properly in order to make it work and insert it into a functional complex so it can actually provide a function for the cell. If it's misfolded, you need to be able to recognize that and degrade it using the proteasome. If it doesn't get recognized, they can then form an aggregate where you get these cell changes of aggregated proteins which characterize neurological disorders such as Alzheimer's and Parkinson's disease. So in Alzheimer's disease, you get an aggregate of beta amyloid, whereas in Parkinson's disease, you get alpha-synuclein. So the cell has to be able to recognize unfolded proteins or misfolded proteins in order to get rid of them so you can get proper cellular function. Now, an essential function of the immune system is to recognize foreign proteins, which are either viruses or bacteria, and get rid of them. So the immune system and the UPS system work hand in hand. And there are two parts to the immune system: the adaptive immune system, which is B cells and T cells, antigen is produced, and it has a memory. Now, this isn't so important to the brain because it's kept out by the blood-brain barrier. What's more important to the brain is the innate immune system, which consists of a series of receptors in your cells that recognize foreign proteins, whether it's viral proteins, whether it's bacterial proteins. It activates the pro-inflammatory cytokines and causes a cascade to get rid of those foreign proteins. And there are two parts of this system. There's the TLR toll-like receptors, and then there's the complement system. And we know in the last couple of years, the complement protein C4 was shown to be important in schizophrenia. Both of these systems, when they get activated, produce pro-inflammatory cytokines such as TNF-alpha or S100 proteins such as S100A8, which I'll come back to in a few minutes. So the innate immune system is non-specific. It acts immediately. It includes anatomical barriers such as the blood-brain barrier, and as I say, the complement pathway and the toll-like receptor pathway as well. And certain receptors occur on different cells of the brain. TLR1 to 8 occur on the brain's immune system, brain's immune cell, microglia, TLR2 and 3 on astrocytes, and we're not quite sure what's expressed on neurons. Parallel to the TLR system, or the innate immune system, is the very important neurodevelopmental pathway called Wnt. And what's been realized in the last few years is the Wnt system, which controls the development of neurons and the development of the brain, actually interacts very closely with the innate immune system, with the TLR, and it modifies the activity of that system. And likewise, the TLR system also modifies the activity of the Wnt system. So both of these pathways interact and are related. And I'm going to show you some of the work we've done showing changes in these pathways, including interferon-related proteins. So this is a study I did a number of years ago where we were looking at an inflammatory protein in the brain called calprotectin. Now, calprotectin has been researched a lot in inflammatory bowel disease. It's a biomarker. And we demonstrated for the first time a number of years ago that it was also expressed in the brain and that it seemed to be expressed by microglial cells. So this is the typical appearance of calprotectin, which is a dimer of two S100 proteins, A8 and A9, and all three of these proteins are expressed in microglia. We showed that in the brain in schizophrenia, the expression of calprotectin was significantly elevated, again, indicating an inflammatory component of this disorder. And then a few years ago, while I was in San Diego, we did a microarray study and found that the one gene that was most altered in terms of high expression was this S100A8. We showed it was increased by fourfold in microarray, and then when we validated this using RT-PCR, it was a fivefold increase in the expression of that inflammatory gene. Again, adding to the idea that this disorder has an inflammatory component to it. Another very important pathway is the tumor necrosis factor, which again is part of that innate inflammatory pathway that gets activated when there is some damage. When TNF is produced by the cell, it's inserted into the membrane, and it's called transmembrane TNF. While it's in the membrane, it can actually interact with two TNF receptors, receptor 1 or receptor 2. While it's in the membrane, it prefers to interact with receptor 2. There's a protein called ADAM17 that comes along and cleaves this, and then you get soluble TNF-alpha. And also that protein cleaves the receptors, so you get soluble receptors. When the TNF is soluble, it now prefers to interact with receptor 1. And the reason why this is important is will come clear in the next slide when I show you some work we did. So we looked at the expression of the components of this pathway in the dorsolateral prefrontal cortex in the brains of schizophrenia, major depression, and bipolar disease. We measured the actual amount of TNF itself, we measured the protein in the membrane, we measured the soluble protein, and we measured the gene expression of receptor 1 and receptor 2. In those brain tissues that we'd got that had major depression in life, there was a massive increase in the expression of TNF-alpha in the membrane. Remember I said this, in the membrane, it prefers to interact with receptor 2, and there was a significant decrease in the gene expression of that receptor. In bipolar disorder, the only change was a significant decrease in the expression of receptor 2. While in schizophrenia, we noticed a very high increase in the expression of receptor 1. I think this was probably the first time where you could actually tease apart the different clinical diagnoses by the change in the expression of this TNF system. We need to replicate this again to see if it's generalizable to other brain cohorts. But again, I showed this merely to show you that there's an inflammatory component to psychosis, and we're trying to still understand the contribution of that to the development of the clinical disorder. While I was working in the University of California, San Diego, I was doing a lot of work there on HIV, and we could follow up people in life who had regular neurological and psychiatric assessments every 6 months or a year, and if and when they died, we were able to have an autopsy and get access to the brain tissue. San Diego, about the time I arrived, was the kind of the ground zero for methamphetamine, and people would go into the desert and they cook it up and then they come back and it was cheaper than alcohol. So I was able to look at the effects of methamphetamine on the brain because we knew from the prospective assessment of individuals how much methamphetamine, if any, they'd taken during life. And this was a cohort of people who were also living with HIV. What we showed is that in those who had used methamphetamine, there was a significant loss of a population of interneurons that expressed a calcium binding protein called calbindin. Now, it took us a whole bunch of experiments to work out it was this particular specific population of neurons. And this is what they look like in a control case, where there's HIV encephalitis, but where there's been meth use in life. And the loss of those interneurons correlated with the last score on a memory score during life. So we knew that losing these neurons had a clinical effect on ability to perform cognitively. We then, having seen that there was a loss of this specific population of interneurons, wanted to work out exactly why that was occurring. And again, we did a whole bunch of experiments, and what we ended up finding was methamphetamine resulted in a significant increase in interferon-related genes, and especially this interferon-stimulated gene called ISG15, 15 kilodaltons, and we showed it was increased by 5-fold. And again, this is what you'd expect to see in your or my brain, where there was some HIV encephalitis, but where there'd been meth use in life, there was a significant turning on of that gene. What I haven't shown you here, but I've got some other slides where we show that the more that protein gets turned on, the more neurotoxicity is, and the more loss of neurons or damage to neurons. So we know that methamphetamine results in an increase in interferon-related genes, and that in itself is neurotoxic. And again, people who take methamphetamine have much higher rates of psychosis. So I was using this as kind of a sort of organic model of psychosis. I want to return to the Wnt pathway, which is the pathway that controls brain development and is also active in the adult brain. And Wnt is an extracellular signal that interacts with this receptor called frizzled, that then activates a protein called disheveled, and then which interacts with this complex, which includes an enzyme called GSK3 beta that regulates the cytoskeleton and synaptic proteins. I don't know why they've got such funny names, but apparently these were named from knockout Drosophila, where if they lost that receptor, the flies look frizzled. If they lost that protein, they look dishevelled. I don't know what the difference is between a frizzled or dishevelled fruit fly, but somebody clearly does. Anyway, we did a series of experiments looking again in the brains of people who had schizophrenia during life, and we found that there was a significant decrease in the expression of this kinase called GSK3 beta. This is a very important kinase. It's the one that goes wrong in Alzheimer's. We showed it goes wrong in HIV dementia, and we know that part of its main function is to maintain the cytoskeleton through the phosphorylation of the cytoskeletal protein MAP2, and also the maintenance of synapses. And you may remember earlier on I said one of the most robust changes in the brain in schizophrenia is lock of markers of dendrites and synapses. We know this is a disorder of connections between neurons. And we also know that that Wnt pathway interacts with the innate immune system. I showed you a slide earlier on with the interaction between the Wnt and the TLR pathway. We and others have looked at this pathway over the years, and there are a number of groups, including ours, who have shown changes in components of this pathway. Here we showed a decrease in GSK3 beta. In that same complex, beta-catenin is decreased, as is the protein APC. And we know that MAP2, the cytoskeletal protein, is also dysregulated in the brain in schizophrenia, as are markers for synapses such as synapsin 1. Interestingly, we were able to show that the expression of some of these genes also correlated clinically with the severity of the SANS and the SAPs. And here's just a few. This is from a blood study we did, looking at the gene, the expression in the blood of the dishevelled gene and also GSK3 beta. And again, showing changes in the expression of these genes correlates with severity of negative symptoms. I think there is some relevance of these changes to the clinical presentation. We know that decrease in GSK3 beta is linked, as I say, to synaptic and dendritic changes. We know that drugs such as amphetamine, LSD and PCP also affect the expression of GSK3 beta. And that pathway also responds to an enriched environment which increases the expression of the Wnt protein which interacts in this pathway. And so we know that this pathway is probably important clinically, and as I said before, it modulates the innate immune system. So going back to what Carmina said about changes in pro-inflammatory cytokines, this pathway can also influence the way in which those pro-inflammatory cytokines are produced. And we know that lithium chloride is an inhibitor of GSK3 beta activity, and also it also affects microglial activation. So if you're still with me, I hope there's not too much technical information so far. And I want to move on to the related pathway. So when you get activation of the immune system and it detects foreign abnormal proteins such as viral proteins, it then activates the ubiquitin proteasome system, and it takes over the proteasome activity, and I'll explain about this in a minute, and it becomes the immune proteasome system. So this is a way of the cell getting rid of foreign proteins. This UPS, or ubiquitin proteasome system, is really important in most functions of the cell. Protein degradation, DNA repair, endocytosis, autophagy. Autophagy is used to break down organelles within the cell, and also it responds to immunity and inflammation. So it selectively targets individual proteins in our cell, which it believes are either should be short-lived, such as cytokine proteins. They express their activity and then should be broken down, or proteins which it senses to be damaged or defectively folded that can't carry out their job properly. This accounts for 8 to 9% of all intracellular proteins. They all have to be monitored, broken down, and recycled. So short-lived proteins that finish their physiological function, such as neurotransmitters or neurotransmitter receptors. After a receptor is activated, that receptor is endocytosed and broken down. If that doesn't occur, memory doesn't get formed. We know that that cycle is now essential to the development of memory. Got to get rid of hormones after they've had their effect. Got to get rid of proteins which don't belong there, such as viral proteins or bacterial proteins. And so all cells need a process to maintain the required amount of protein for normal cellular function. This is proteostasis. And this is just some of the functions that the UPS system is involved in. Inflammation, MHC1 antigen presentation, what I call garbage disposal, things which shouldn't be there, need to be got rid of, keep the cell clean. And we know that this system goes wrong in neurological disorders with the deposition of aggregate proteins, amyloid in Alzheimer's, also tau in Alzheimer's, alpha-synuclein in Parkinson's disease, and TDP43 in frontotemporal dementia. So this pathway goes wrong a lot in neurological problems, but no one had really looked at it to see if it goes wrong in psychiatric disorders such as psychosis, which is what we ended up doing. So we'll just explain the process to you. It's really quite simple. So you've got a protein here that the cell has decided is damaged or is end of its life, and so it's got to get rid of it. There are 3 sets of enzymes called E1, E2, and E3. And what these 3 enzymes do is take ubiquitin and transfer it onto that protein, and you get this ubiquitin chain, and that then is a tag to say, break this protein down. A bit like if you park your car illegally and the traffic warden comes and puts a tab on it and says, take this car away. So this ubiquitin chain says to the cell, this protein has got to be got rid of, so it then is transferred to the proteasome, which is the cell's shredder or vacuum cleaner. The protein gets shredded, the ubiquitin is taken off, and then it all is recycled again to break down another protein. So there's this whole process of adding ubiquitin to a protein, the proteasome breaking the protein down, and then a series of deubiquitination enzymes taking the ubiquitin off so it can go into another cycle. Clear? You understand that? Good. If it goes wrong, this is what you get. This is my son's bedroom a day after it was cleaned up. So the UPS is trying to get rid of things that shouldn't be there, like discarded trousers and plastic bags, and can probably clear those up and so try and keep this bedroom as clean as possible. But there are certain things which get too big, and these become deposited aggregated proteins, such as amyloid, synuclein, or TDP43. So it's important to keep that bedroom, that cell, clean all the time, otherwise things happen you can't get rid of. We just hope they grow up. What I'm not going to talk about, there are two other systems in the cells that can get rid of proteins, but normally larger things like organelles, and this is autophagosomes and lysosomes. But I'm going to concentrate today on the UPS system because I don't want to confuse you even more. So is there any evidence for this pathway being abnormal in schizophrenia? Well, Mystery did a GWAS study of 153 patients with schizophrenia and 153 controls, and they looked at the gene expression, the top categories, and they found that one of the top categories was ubiquitination and neurotransmitter transport and neuroreceptor transport. Again, that transport is heavily dependent upon the ubiquitination system for it to work. There was a GWAS study that I was involved in when I was at UCSD. Again, we looked at 107 cases of schizophrenia, 118 controls, and we found negative control of biological processes, just GSK3 beta, and then also expression of interleukins and other cytokines. And as I say, a five-fold increase in that inflammatory protein S100A8, which I showed you earlier on when I was describing the work we did on the inflammatory protein calprotectin. Another study using laser microdissection, where you put the microscope over a cell and then the laser just cuts it out so you can actually work out what is being expressed in individual neurons. Again, they found that UPS-related genes were more prominently expressed in neurons in layer 5 in schizophrenia. And then Jerome Breen at the Institute looked at meth psychosis compared with non-meth psychosis, and again found that it could differentiate the two groups using genes in the ubiquitin proteasome system. So there's some accumulating evidence shows that this pathway would be going awry in schizophrenia. While I was at UCSD, we did a GWAS study comparing gene expression in the brain and blood in schizophrenia, and although there are 20,000 proteins or genes, only 6 were dysregulated in both the brain and the blood, and of those, only 1 was dysregulated in the same direction in both compartments. So selenium-binding protein was a gene that was increased in expression in both the brain, the dorsolateral prefrontal cortex, and the blood in schizophrenia. And this is one of the first pieces of data that got me interested in the UPS, because while we're not, I'll come on to the functions in a bit, and we then replicated this in 2 other brain collections, one from the Stanley and when I was in Melbourne from the Victorian Brain Bank collection. And again, in all 3 cohorts, we found increased expression of selenium-binding protein gene in different regions of the brain in schizophrenia, and it didn't occur in affective disorders, either bipolar or major depression. And at the protein level, this is the expression of selenium-binding protein, tend to be in glial cells, whereas in schizophrenia there's more prominent expression and could possibly be in neurons as well. We're not quite sure what selenium-binding protein does. We know it binds selenium. It's not a classic selenoprotein, though, but we know that it has a role in ubiquitination, and it's clearly linked to ubiquitin-related genes and an S100 inflammatory protein. So looking at the expression of this gene, this selenium-binding protein, got me interested in looking further at the ubiquitin proteasome system. And we did a study where we looked at 4 different cohorts, 2 in San Diego and 2 in Taiwan, schizophrenia and bipolar disorder, and we looked at the gene expression pathways, and we found that there was one pathway that was common to all 4 cohorts, and that was the protein ubiquitination pathway. And that some of the genes in that pathway, again, correlated with the severity of clinical symptoms. So the ubiquitin conjugation genes correlated positively with symptoms on the SAPS. So this is SAPS, not SANS. The GSK3 beta was with negative symptoms, this is positive symptoms. And then one deubiquitination gene correlated negatively. So here's just showing, so the higher the expression of these ubiquitination genes, the higher the SAP score, whereas the deubiquitination gene, the higher the expression, the lower the SAP score. More recently, before I left Melbourne, I had a great PhD student, Chenxing Liu, from Peking University, where we did a joint study looking again at a GWAS, looking at an African-American population, a European-American population, and a Han Chinese. And although there were lots of different pathways which were dysregulated in these three cohorts, only 4 pathways were common to all 3 ethnic groups. And in that 4 pathway was, again, the ubiquitin-mediated proteolysis. So again, ubiquitination is appearing quite prominently. Now, it's all very well having these gene expression studies, having these correlations. What about actually looking at the activity of the pathway itself? This is more difficult to do. And I'm going to talk about work we've done in a few minutes. So Rubio's lab looked at the expression of the UPS in the superior temporal cortex in schizophrenia and found a reduction of protein ubiquitination, of free ubiquitin, and of a particular type of ubiquitination, which I wouldn't worry about. And again, this was also found in a group of elderly population of schizophrenia, but no effect of the drugs. And we know, as I said earlier on, that as soon as transmitter is released and binds to a receptor, that receptor is endocytosed and it's broken down by the UPS system. And if that doesn't occur, then whatever memory or activity should occur will not occur. So the release of neurotransmitter will have been in vain. This has to be endocytosed and broken down by the UPS system for the activity to occur. We know that there are, so the proteasome activity can be inhibited, and there are medications already available which are used in cancer, and that's what they're called. And then, just over a year ago, Scheibe did a case report of 5 individuals severely affected with NMDA encephalitis who required intensive care ventilation. They were unresponsive to steroids, intravenous immunoglobulins, immunoabsorption, or cyclophosphamide. But they all were improved when they were given a proteasome inhibitor. So I worked with a psychiatrist who already got in his lab assays to measure the activity of the UPS system. So together we looked in the brain and the blood in schizophrenia, measured the amount of free ubiquitin, we measured the amount of protein that you ubiquitinated, we measured ubiquitination activity and the proteasome activity. This is just the description of the cohort. When we exposed some mouse neurons to clozapine, we showed no effect on the UPS system. So we know that this is not due to drugs. But when we looked at both the blood and the brain, there was a buildup of ubiquitinated proteins both in the blood and the brain in schizophrenia. So more proteins are being ubiquitinated, but they're piling up. There was a decrease in ubiquitination activity in the blood, but no change in the brain. So even though more proteins are being ubiquitinated and piling up, the actual ubiquitination activity is slowing down. And there is no change in the proteasome activity either. So both the blood and the brain in schizophrenia is characterized by a buildup of these ubiquitinated proteins, which the cell is not getting rid of. So this is my kind of final slide, really. The UPS system, I've only just scratched the surface in terms of describing the activity of that system is heavily dependent on metals, especially iron. And we know that in the brain, iron is stored by ferritin, and that amyloid precursor protein, the protein which goes wrong in Alzheimer's disease, promotes a neuronal iron export. Iron activates tyrosine hydroxylase, which is the enzyme necessary to produce dopamine. So that enzyme will not work unless iron is around. And alpha-synuclein inhibits that enzyme and also inhibits dopamine release. most recently, we have shown these findings, which are still in the labs, have not yet published. We've shown that in the brain, there's an increase in the expression of iron. There's no change in the iron storage protein, ferritin. There's also a decrease in APP levels and alpha-synuclein. So iron is increased. That may be leading to an overactivity of tyrosine hydroxylase and therefore more dopamine production. And the two things that can inhibit that, alpha-synuclein is decreased, and also APP, which promotes neuronal export, is decreased as well. So the UPS system may be driven by changes in iron, and we're looking further at this anyway. This is, say, data that's just coming out the last couple of weeks. So, in summary, we've shown there's a build-up of ubiquitinated proteins in the blood and the brain in schizophrenia. No change in prosome activity, ubiquitination may be decreased, and there are also changes in components of the innate immune system with cytokines such as ISG15, tumor necrosis factor alpha, and the Wnt pathway. And there is a very enmeshed relationship between the innate immune system and the activity of the proteasome. I say we're now looking at the relationship with the activity of that system, the UPS, on dopamine synthesis. So I hopefully have convinced you that an element of schizophrenia includes abnormalities in proteostasis or the alteration of cellular cleanliness. And I think I'll stop there. This is just a factory in Dresden where they make VW cars, which is called the Transparent Factory. And it is the most clean factory I've ever seen in my life. And if I wanted to have a model of what a clean cell looks like, it would look like this factory. Unfortunately, oddly enough, they call their luxury car the Phaeton, which was a very popular open carriage in the 18th and 19th centuries, drawn by one or two horses. But it's also the name of the son of the sun god, who convinced his father to let him drive the sun chariot, and he couldn't control it, and so he was shot down by Zeus in order to prevent a disaster happening to the world. So I think it's an odd name to give to a car that can actually cause disaster. Thank you very much.